Biomedical research has historically relied very heavily on the use of animal models and in vitro models. However, both approaches have shown to have very severe limitations. If we think about animal research, limitations are mainly for ethical reasons, and if we think about in vitro research, is because not all the research that has been generated using these models has really a high translational value for therapeutic reasons. Therefore, we have seen that in the last few years there has been a very great development of what are called new alternative methodologies, which combine advanced cellular models with advanced technologies in order to generate what are called organoid models.
The organoids that we are generating here in EURAC research are basically three-dimensional cellular models that are generated by human cells, so are human cellular models. So we are not generating an entire organ, let's say an entire brain in this case, but we are generating, creating different part of the brain, single different part of the brain that we are able to study and to analyze.
This is a great tool for the future because these models allow us to generate more knowledge, more chances of cure disorders which have a high impact for patients and the families and society.
In our lab we are studying neurodegenerative diseases and mainly Parkinson's disease. To this aim, we are generating two types of organoids, of brain organoids. One that is similar, let's say, to the cerebral cortex, and the other that is similar to the substantia nigra. These two parts of the brain are important in the study of Parkinson's disease because these two parts are the more affected during the disease. So our main goal in the lab is to develop these models, study these models, trying to understand how the cells that are present inside are working and what is the main difference between the physiology and the pathology of the disease.
Brain organoids generated at EURAC are then combined with different types of chips. These chips are made of different types of polymers, such as plasticopolymers, and they can contain sensors as well, such as pH, temperature, oxygen, or as in case of the chip we use here, they contain microelectrode arrays. These electrodes can measure the electric activity of neurons in 3 dimensions and in such a way provide us with very detailed information about the functions and dysfunction of such brain organoids.
In our lab, we are analyzing our brain organoids in terms of morphology and also activity. To study the morphology, which is the organization of the tissue and shows how the neurons and the cells inside the tissue are positioned, we are using the microscope, so the confocal microscopy. Instead, for the analysis of the activity, we are recording the organoids, so we are taking the organoids and place on a chip, let's say, and that is able to register the electrical activity of the tissue.
This line of research is also following a worldwide movement for the development of such alternative methodologies. In fact, also in the United States, the FDA has released new guidelines for the development of methodologies which can finally replace completely animal studies for the approval of new therapies. Such Modernization Act, in fact, is aiming to integrating advanced cellular models, advanced technologies, and finally develop complex organoids which can create organs which are specific for different tissues.